Foraging involves searching for food, and feeding results after a food item has been discovered and accepted as a suitable plant part, prey item or host of a parasitoid. The process appears to be simple enough for humans, but for small insects with specialized diets, and a complex environment in which to search, the adaptations that overcome such challenges become complex. This point is illustrated by the path of behavioral decisions made by a parasitoid from finding an appropriate habitat to host acceptance and oviposition, and manipulation of the host. While some specifics are provided here, the scheme of decision-making resulting in the narrowing of the field of search may be generalized to the sequence of behaviors that the majority of insects must pass through in order to find food. This is the case whether an individual is finding food for itself or for its progeny. One of the most thoroughly studied species regarding foraging behavior is the parasitoid, Cardiochilus nigriceps, Hymenoptera, Braconidae, which uses as a host the tobacco budworm, Heliothis virescens, Lepidoptera, Noctuidae. The species has been studied over many years in Vinson's research group, with a detailed flowchart of interactions resulting, as shown in Figure 2.3, Vinson 1975, de Morais et al. 1998, de Morais and Lewis 1999. The remarkable number of behaviors involved in the progression from finding a suitable habitat in which to search, to finding a host and accepting it, and ovipositing in the host, is well illustrated in the figure. First, females must locate a suitable habitat, one in which the host is likely to be found, and they are attracted to brightly lit, open areas in which tobacco plants may be growing. Once in the habitat, 2, females search for host plants, 3, and examine them for damage, 4. They land on plants that are damaged by tobacco budworm larvae in response to a host-seeking stimulant, 5. This chemical, a mixture of 3-methyltriocontanes, originates in the mandibular gland of the host caterpillar, which then becomes deposited on leaf surfaces as the caterpillar feeds. The parasitoid, on detecting this compound, 6, starts actively searching the leaf surface, with antennae pressed against the leaf. She follows the path off the compound until she finds a host, 7. The mandibular gland secretion that provides an adaptive advantage to the receiving parasitoid, but is produced by the individual the parasitoid is searching for, is called a chiromone, see Table 2.3. Once the host caterpillar is discovered, the host acceptance stage in the sequence of behaviors is reached, 8, Figure 2.3. A specific host compound on the cuticle appears to provide a stimulus for oviposition, although the host may be rejected if it has been parasitized previously, based on a host marking chemical left by the original female. A previous parasitoid attack also leaves an internal repellent to subsequent oviposition, which is detected by a second female once the ovipositor is inserted below the cuticle. This results in rejection of that host. However, if the host is accepted, oviposition follows and the host regulation phase of the interaction develops. 9. With the egg are injected substances that enhance larval parasitoid survival, modifying physiological processes in the host that benefit parasitoid fitness. This sequence of stimulus-response behaviors is remarkable in its complexity. However, it is no doubt representative of the kinds of interactions when insects are searching for food, be they parasitoids, predators or herbivores. At the contrasting scales of large complex environments and small insects, many adaptations for finding food have evolved using chemical communications. Often, it is simply the body odor, BO, of the host plant or insect, which acts parasitoid and host ecologically and chronologically coincident. As a stimulus, or odors from particular glands, Scales or Frass, Price et al. 1980, Price 1981. See also Chapter 13 on multitrophic interactions. In addition to using innate responses to stimuli, insects can learn to find food more efficiently, Papage and Lewis 1993. Parasitoids learn to associate the discovery of hosts with odors emanating from food, such as from yeasts or apples, associative learning. They can even learn to associate hosts with chemicals exotic to their environment, such as perfumes, vanilla and chocolate. Flower-visiting insects learn to improve their access to rewards in individual flowers with experience, and some may learn routes along which nectar or pollen can be found, trap lining. The local path followed by a searching predator or parasitoid has attracted much attention, partly because it reveals the efficiency of a potential biological control agent, and partly because the rate of discovery affects the population dynamics of the natural enemy and prey or host interaction. The question most simply put is how much time should a searching predator, or parasitoid, spend in a patch before giving up the search? 3 scenarios for this patch time allocation have been proposed, e.g. Van Alphen and Vet 1986. 1. Number expectation in which an enemy leaves a patch after finding a certain number of hosts. 2. Time expectation in which an enemy leaves a patch after a fixed amount of time. 3. Giving up time in which the enemy gives up searching and leaves the patch if it has not found a suitable host within a fixed time. The number expectation scenario is supported by Cardiochilus nigriceps, which leaves a patch after successfully attacking one host. But, in general, the number of cues that affect a search pattern are too numerous for any simple model to apply effectively. The ichneummonid parasitoid, Venturia canescens, searches for its host, the Indian meal moth, Plodia interpunctula Lepidoptera, Pyralidae, which feeds on stored grain and meal, and spins a web as it moves through the medium. The wasp is attracted by the silk and turns frequently after contact, Figure 2.4, remaining in the patch until the stimulus is no longer effective. But if a host is found and oviposition occurs, then the parasitoid increases its sensitivity to the patch edge, thereby continuing to search within the patch. Therefore, time spent in a patch depended very much on the number of hosts discovered in that patch, but the time in the patch did not correlate with host density, or with expectations based on the fixed giving up time model. With with so many variables relevant to a searching parasitoid, e.g. Wanberg 2006, inevitably significant interactions among factors are likely to occur, making the discovery of patch residence rules more problematic, Muratori et al. 2008. Van Alphen and Vet 1986. See also Wanberg 2006, Wanberg et al. 2008 list a series of factors which may influence patch time allocation, emphasizing the need to know their influence and Figure 2.4 The search pattern of a female parasitoid, Venturia equals Nemeritus canescens, on a glass plate, within a patch of host secretions. The edge is marked by stippling in the figure. Arrows show where the female turned abruptly at the edge of the patch. From Wahage 1978. Relative importance before an adequate patch time model can be formulated. 1. Patch structure, for example, patch size and roughness. 2. Chiromone concentration, as concentrations. Increased patch time is likely to increase. 3. Unparasitized hosts in a patch increase patch time as oviposition takes time and often stimulates more intensive search. 4. Parasitized hosts decrease the likelihood of a female staying in a patch because rewards decline. 5. Other parasitoids, either con or heterospecific, may leave trail odors which are repellent to an arriving female. 6. Previous experience in other patches may reduce patch time, including when mixed host patches have been experienced. 7. Travel time between patches probably increases. The patch time as motivation to search increases. An additional factor that has become increasingly realized recently is the importance of plant-provided food, nectar, pollen, sap, extrafloral nectaries, etc. So that patch dynamics is strongly influenced by alternative foods to prey because predators and parasitoids are frequently omnivores. Eubanks and Stiersky 2005, Van Rijn and Sabellis 2005, Wa Diaresis Kurz et al. 2005. When considering predators, many of the factors remain the same as for parasitoids. However, whereas many parasitoids attack slow-moving or stationary hosts, predators may cause a scattering of prey individuals. See 2005. When this occurs, such as when backswimmers, Hemiptera, Notonectidae, attack mosquito larvae which have escape responses, the notonectid's preference for high-density prey is offset by an increased scattering of mosquito larvae, resulting in no correlation between prey and predator densities. In addition, if mosquito larvae benefit from a predation refuge, a negative correlation between prey and predator results. Nevertheless, most modeling has assumed stationary prey, applying optimal patch use approaches or the concept of ideal free distribution studies. The former emphasizes the individual's response to prey and the latter the population response. The ideal free distribution assumes a set of habitats or patches that differ in quality, e.g. prey density, and the predator is free to search, having an ideal knowledge of patch densities of prey. Predators will then show an aggregative response to prey density, causing prey density in the patch to decline, after which predators will find other patches more profitable. Eventually then, the predator population is distributed such that each individual is acquiring food at the same rate, a stable ideal free distribution. Such distributions have been observed in predator-prey, parasitoid host and herbivore-plant interactions. For example, one of a small number of unsimplified studies under natural conditions shows an ideal free distribution for the gall-inducing aphid, Pemphigus vitae, selecting breeding sites according to leaf size of the host plant, Populus angustifolia, Witham 1980, 1981. The mechanisms involved include females jousting for the best locations and are discussed in Chapter 4 in subsection 4.3.9, spatial and temporal variation in plant allochemistry and nutrition pose tracking problems for insect herbivores, and are illustrated in Figure 4.11. Obviously, foraging and feeding are large topics which will be covered in other parts of the book as well, particularly where food is toxic or of low nutritional status, Chapter 4, where there is competition for food, Chapter 5, when symbionts open up new resources, Chapter 6, when feeding and host specificity of parasites is considered, Chapter 8, and when community structure and multitrophic level interactions are discussed, Chapters 12 and 13. Indeed, much of ecology concerns the behaviors involved with food, feeding and foraging. 2.6a Place to live. Just as with finding food, every individual of every species, or its mother, has to find a place to live if it is to survive, so the subject of habitat and microhabitat selection and use is of general interest in ecology. How an insect navigates a complex, heterogeneous locality to find a profitable place to live, and the cues that elicit appropriate behaviors relating to habitat use, are enduring questions, with many kinds of adaptations and strategies involved. Some of the diverse major categories concerning where insects live are listed in Table 2.2, and are divided into distinct types of answers to the challenge of occupying a safe place to live. Some examples include 1. Females oviposit exactly where the larva will begin to feed. 2. Females construct or induce habitats for progeny. 3. Females provision cells in which larvae feed. 4. Plants provide microhabitats protective for insects and others. Table 2.2a Place to live and care for progeny. Some of the microhabitats in which insects and other arthropods live. 1. Females place eggs in favorable sites for larvae. Preference performance linkage, e.g. Pemphigus aphids, Urosophiles, Bruchid weevils and many others. Price 2003a. 2. Females make habitats for progeny, e.g. galls, leaf folds and rolls, nests of bees, wasps, ants, termites, dung beetles, carrion beetles. 3. Provisioning, usually by females including sterile workers, for larvae in concealed places, e.g. cells in nests, dung balls, carrion fed to sylphid larvae in nest, a subset of 2 above. 4. Plant provided microhabitats, e.g. domatia for predatory mites. Myrmecodomatia, hollow stems, abandoned galls, flaky bark. 5 animal-provided habitats, e.g. pelts of mammals, feathers of birds for parasites, noses, ears, underskin, blood, organs, etc. 6 weaving tents, webs and cocoons, e.g. webworms, tent caterpillars, web-weaving spiders, nest-weaving ants, silken cocoons for pupae. 7 burrowing in substrates. A. Into dead or dying wood, passolid, leucanid, cerambycid and buprested beetles, carpenter bees, carpenter ants, termites. B. Into ground, scarabaeid beetles, crickets. Elaterids, ants, solitary wasps. C. Into living plants, larvae of many herbivorous insects, e.g. curculeonid and bruchid weevils, gall formers, leaf miners, fruit feeders. D. Into living animals, parasitoids, some bot. Flies, chiggers, scabies. Mites. E. Into sand, pebbles, or under rocks, litter or other organic debris, e.g. larvae of some moths, spiders, beetles, aquatic insects such as caddisflies. 8. Abandoned oviposition, i.e. oviposition away from a safe feeding site for larvae, e.g. some arctiid moths and stick insects drop eggs to the ground. Many moths oviposit onto substrates largely irrelevant to where larvae will feed, rocks, tree trunks, leaf litter. Therefore larvae lead a dangerous, exposed life until they establish a feeding site on a plant. 9. Feeding in open, e.g. many free-feeding herbivores, tenebrionids, carabids, many other predators. 10. Living in or on water, e.g. water striders, coryxids, brine flies, black fly, mosquito larvae. 11. Urban environments, e.g. roaches, spiders, ants, termites. 5. Animals provide microhabitats for parasites. 6. Weaving insects and spiders make nests for themselves. 7. Burrowing by adults and larvae into substrates. Provides shelter and food. 8. Indiscriminate oviposition behaviors. 9. Feeding in the open. 10. Living in or on water. 11. Living in urbanized environments. It is hardly surprising that, for small organisms, they can be found almost everywhere. These examples are discussed in more detail below. 2.6.1 Females oviposit exactly where their nymphs or larvae will begin to feed. Table 2.2, 1. A female that is highly selective in oviposition behavior shows a strong ovipositional preference for a certain quality of resource for its progeny, among a range in resource variation. Females have evolved to be specialists, and show a remarkable capacity in a complex environment to make excellent choices. We can judge a female's quality of choice only by evaluating the performance of her progeny. Thus we look for a high ovipositional preference, larval performance linkage. Larval performance is often measured as the probability of survival, or growth rate. Such linkage is relatively straightforward to measure in gall-inducing insects because the gall is initiated by a female in some cases, showing her preference for a particular site, and the progeny's performance is evident within the gall. For example, stem mothers of the aphid, Pemphigus vitae, are first instar nymphs, only about 0.6 millimeters in length, and they must crawl from a tree trunk into the foliage of a large tree to initiate a gall in which they and their progeny will feed. Witham 1978, 1979, 1980. They show strong preference for the largest leaves on a tree, figure 2.5, and they joust for the best position on the largest leaves. At these sites they are highly successful at initiating galls and producing more progeny than on smaller leaves, see figure 4.11. A very similar preference performance linkage is shown by the shoot-galling sawfly, Eurolaciolepis, in which long shoots are preferred as oviposition sites by females, and larvae survive best on these shoots, figure 2.6, Craig et al. 1986, 1989. These kinds of examples promoted the development of the plant vigor hypothesis, which argues that many insect herbivores select and perform best on plants and plant modules which are growing vigorously in a population of plants or modules. Price 1991a, 2003a. See also Chapter 4 on the plant stress and plant vigor hypotheses. 31.8 1.6 1.4 1.2 1.0 0.8 0.6 0.4 0.2 25 20 10 LEAVS PERAVIL LEA dash. Figure 2.5 The availability of leaves in leaf size classes, solid circles and solid line, in a narrowleaf cottonwood tree, Populus angustifolia, and the choice of stem mothers of the aphid, Pemphigus vitae, solid bars and dashed line. Note the strong preference for the largest leaf area classes, which are the least abundant classes in the leaf population. From Witham 1981. 2.6.2 Females make habitats for progeny, table 2.22. In the gall inducer examples above, the young female aphid stimulates gall formation and she and her progeny live in the gall. The adult female sawfly oviposits into plant tissue, stimulates a gall, lays an egg, and the larva feeds within the gall. Other structures can be induced by ovipositing females such as leaf folds and rolls in which their larvae feed. Major nest builders include bees, wasps and the termites. Some carrion beetles also construct a nest under the carcass they have buried, and feed their larvae rather like birds feeding chicks in a nest. Dung beetles construct dung balls in which they lay an egg, and the larva feeds within the ball, see figures 2.17, 6.17a, b. 2.6.3 Females provision cells in which larvae feed, table 2.23. Provisioning larvae in cells is a general trait in many social insects and solitary wasps, as we discussed in section 2.3 and illustrate in figure 2.1. We provide much more detail on provisioning in chapter 3 on social insects. 2.6.4 Living plants provide microhabitats, table 2.24. Insects and other arthropods can search for and find many microhabitats on plants which provide protection and food. This is illustrated in the oak tree example, figure 1.3, and the typical plant example, figure 4.1. The text associated with each of these figures provides a little more detail on these kinds of microhabitats. 2.6.5 Living animals provide microhabitats, table 2.25. Parasites live in and on animals just as on plants. An example of the many microhabitats that can be used is given in figure 8.1, showing human skin and all the places occupied by mites. Humans also provide head, body and pubic lice a habitat to occupy, and chewing lice on other animals usually specialize on a 25, 20, 15, 10, 5, 0, 0, 40, 80, 120, 160, 200, 240, 280, 0, 20, 40, 60, 80, 100 galled shoots. All shoots. Percent. G. A. L. Ed. Percent. O. F. T. O. T. A. L. S. H. O. O. T. S. 1983 shoot length, Figure 2.6 The availability of shoots, in shoot length classes, on clones of Arroyo willow, Salix laciolepis, open circles, and the attack by females of the gall-inducing sawfly, Eura laciolepis. Note that the strongest preference is for the longest shoot length classes, which are the rarest in the shoot population. From Craig et al. 1986. Particular part of the body of a vertebrate host so that several species may coexist on one host. 2.6.6 Weaving insects and spiders produce protection for themselves, table 2.26. Webworms and tent caterpillars construct protective defenses from silk, while many insects tie leaves together. Leaf-tying lepidopterans roll leaves or flatten leaves together and feed within the protected area. Weaver ants in the genus Oecophila pull leaves together and use larval silk to construct a nest of tied leaves. 2.6.7 Burrowing into substrates, table 2.27. The designs and behaviors of burrowing insects is a diverse and fascinating subject. The burrowing way of life is arguably the most common behavioral strategy in the insect world in relation to a place to live. Insects seem to burrow into everything, including substrates not listed in table 2.2, construction timber, furniture, antlers, horns, turtle carapaces, dead skin, dung, carcasses, sea wrack, seaweed washed onto beaches, and all kinds of other places. One prodigious burrower bores down vessel cells of birch trees from the top shoots to the bottom of the trunk, up to 15 meters, then it emerges and burrows into the ground to pupate. This maggot, the birch cambium fly, Phytobia beetli diptera, a gromyzidae, is slim enough as a larva to worm its way down the cambium of host trees, Wylioha et al. 1999. 2.6.8 Indiscriminate oviposition, table 2.28. An interesting example of unselective oviposition is seen in many stick insect species which drop eggs from vegetation to the ground. But ants may collect eggs and carry them to their nests so that nymphs emerge in a safe place, even though away from plant food. Some moth species are known to oviposit on rocks, or tree trunks, even vehicles, and will oviposit in brown paper bags when given the opportunity. Some also drop eggs whilst in flight. 2.6.9 Feeding in the open, table 2.29. Another very common place to live is as a free-feeding insect in the open, the strategy of many herbivores, predators and adult parasitoids. Many adult insects fly to find substrates on which to feed or lay eggs, fruits, seeds, flowers, dung, carcasses, living plants or animals, and so on. Living in the open water provides some protection against terrestrial predators, but there are many predatory species in fresh water from which to hide. 2.6.10 Living in or on water, table 2.210. This category covers many microhabitats mentioned above for terrestrial habitats. Similar behaviors function well in all kinds of environments. 2.6.11 Urban environments, table 2.211. Again, this heading acts as an umbrella for the diverse ways in which insects find or create a place to live, mentioned above. This category reminds us that people have constructed houses, bridges, drains, sewage systems, canals, ponds, lawns, golf courses and gardens, many of which provide ideal places in which insects can live. We can appreciate that there must be a great diversity of behaviors associated with a place to live, all relevant to the ecology of the interaction of the animal and the resource which is utilized. Interesting to contemplate are the details of the stimulus-response sequences needed in each case to search, occupy, construct and live, or lay an egg, in any particular place by any particular insect, as, for example, set out in figure 2.3 for a parasitoid finding a position for its progeny. Or, for a migrating butterfly, finding food, nectar, for itself and for its progeny, e.g. figure 2.2, or the bee-killer wasps, figure 2.1. Such detail is beyond the scope of this chapter even if it were known, so we will progress to the next important set of behaviors in the life of insects. 2.7 Communication. Communication is the act of transmitting information from a signaler to a receiver. A stimulus-response interaction is involved. Some treatments of communication restrict the subject to cases in which signaler and receiver benefit, and both are behaviorally involved in the interplay, e.g. Smith 1977, Greenfield 2002. In books on behavioral ecology, communication has received little attention, e.g. Klopfer 1962, Morse 1980, or the emphasis is on signals that benefit the emitter, e.g. Dawkins and Krebs 1978. Krebs and Davies 1978. The bias toward benefits to one or both participants serves behaviorists well by limiting the subject of communication largely to within-species messaging, courtship and other sexually related behaviors, pheromones, aggressive signals, displays and social cohesion and integration. However, for the insect ecologist the subject of communication is much broader than this view because communication can be active or passive. For example, a beetle may actively defend itself by ejecting a noxious spray when attacked, or a caterpillar may leave secretions on a leaf while feeding that provide a cue for a parasitoid searching for the caterpillar, a passive, non-adaptive form of communication. Here we will concentrate on communication via chemical signals because for insects they play such a critical role in life, for survival, and for finding food and mates. We will examine the many types of communication and then see how they play a role in food webs working up the trophic system from communication from plants to herbivores, then within the herbivore trophic level, and on to communication between herbivores and carnivores. 2.7.1 Chemical communication. Taking chemical communication as an example of the diversity of pathways used in a typical community, plants may communicate passively, by releasing volatiles, which can be considered as body odors BO, and such chemicals may become attractive to herbivores, or natural enemies of herbivores Price et al. 1980, Vet and Dick 1992. An insect may produce strongly repellent chemicals against a would-be predator or parasitoid, and actively reject them at the time of attack, as does the bombardier beetle illustrated later in this chapter. The treatments of communication behavior generally omit consideration of plants as communicators and interactions among individuals from different species, topics of central concern for the insect ecologist. Floral odors are a major component in pollination biology and phytochemicals communicate various kinds of signals, depending on which species the receiver is. In order to discuss chemical communication relevant to insect ecology we need a specialized lexicon on interactions. Terminology follows Nordland, 1981. Semiochemicals are chemicals that mediate interactions between individuals of the same or different species, literally they are signaling chemicals. These may be divided into chemical communication among individuals of the same species, pheromones, or among individuals of different species, allochemicals, Table 2.3. Pheromones are used in different ways, as sex pheromones, alarm pheromones and epideictic pheromones. An epideictic pheromone stimulates dispersal from overcrowded conditions, or from conditions that are likely to become overcrowded, thereby causing a more expanded population structure, Prokopi 1981. Allochemicals are those that mediate interspecific interactions and are divided into 4 classes. 1. Allomones are repellent chemicals which benefit the producer, but are detrimental to the receiver, such as toxic phytochemicals, or stings of bees and wasps. 2. Chiromones benefit the receiver rather than the emitter, for which they are detrimental. This class includes body odors of plants or insects which stimulate searching and attack by their enemies. 3. Cinnamones are those chemicals that are beneficial to the emitter and the receiver, such as plant odors that attract predators of herbivores, or floral odors attractive to pollinators. 4. Apnemones form another important category of semiochemicals in which a non-living substrate emits attractive odors beneficial to individuals responding to the odors, but detrimental to another species in the medium. Parasitoids attacking host larvae in carrion, dung, meal or grain may use the odor of the substrate while searching for hosts. All these allochemical terms relate to interactions involving finding food. This means that different trophic levels are involved. Given the evidence that natural enemies may find herbivores by responding to plant odors, clearly at least 3 trophic level interactions are involved. Price et al. 1980. 2.7.2 Multitrophic level interactions. The term multitrophic level interactions is now generally used to discuss the many ways in which species interact in a food web or interaction web, e.g. Charnke and Hawkins 2002, Oguchi et al. 2007. The subject is much more extensive than just chemical interactions, but it was the study of phytochemicals that gave the subject its initial research energy, e.g. Sondheimer and Simeone 1970. By 1981 the subject of semiochemicals could be treated in a comprehensive way, e.g. Nordland et al. Table 2.3 Definitions of terms used for types of signals in chemical ecology. Hormone, a chemical agent, produced by tissue or endocrine gland, which controls various physiological processes within an organism. Semiochemical, a chemical involved in the interaction between organisms. Pheromone, a substance that is secreted by an organism to the outside and causes a specific reaction in a receiving organism of the same species. Allochemical, a substance that is significant to organisms of a species different from its source, for reasons other than food. Is such allomone, a substance produced or acquired by an organism which, when it contacts an individual of another species in the natural context, evokes in the receiver a behavioral response or a physiological reaction which is adaptively favorable to the emitter, but not to the receiver. Chiromone, a substance produced or acquired by an organism which, when it contacts an individual of another species in the natural context, evokes in the receiver a behavioral or physiological reaction which is adaptively favorable to the receiver, but not to the emitter. Cinnamone, a substance produced or acquired by an organism which, when it contacts an individual of another species in the natural context, evokes in the receiver a behavioral or physiological reaction which is adaptively favorable to both emitter and receiver. Apneumone, a substance emitted by a non-living material that evokes a behavioral or physiological reaction which is adaptively favorable to a receiving organism but detrimental to an organism of another species which may be found in or on the non-living material. From Nordland 1981. 1981, and multitrophic level interactions were emphasized by Price, 1981, who provided references in the literature for the following examples. Interactions between trophic levels 1 and 2, the plants and herbivores, are very extensive, including attractants, repellents and antiphedants, figure 2.7. For example, specialist insects on crucifers Brassicacea are attracted to mustard oils, or glucosinolates, released from potential hosts, also providing the characteristic odors humans associate with cabbage, radish, mustard and other members of the cabbage family. Glucosinolates are attractive to the major species of the herbivore trophic level on crucifers, Luda and Mole 1991, figure 2.7, interaction 1. The chemical structures of a glucosinolate and a mustard oil are illustrated in figure 4.9. In the diptera, the gall midge, Doscinera brassicae, and the cabbage rootfly, Delia brassicae, are attracted to allylglucosinolate. The specialist beetles on crucifers are also attracted: the red turnip beetle, Entomocellus americana, the curculeonid seed weevil, Pseudorhynchus assimilis, and flea beetles in the genus Philotreta. Lepidopterans such as the large white butterfly, Pieris brassicae, and its congeners, P. rapi and P. oleracea, which are more generalist feeders, are stimulated by glucosinolates to lay eggs. However, for other generalist feeders, glucosinolates act as repellents, figure 2.7, interaction 3, including aphids, grasshoppers, lepidopterans and mites not specialized as crucifer feeders. Mustard oils. Trophic. Level 4 Predator SP. 1. 26. 22. 1517 1611. 13. 14. 12. 18. 15. 10. 12. 11. 44. 119. 22. 131. 9855. 76. 12. 25. 21. 24. 20. 23. PARASITOIDSP. 2. PARASITOIDSP. 1. PARASITOIDSP. 1. Associated parasitoids. HYPERPARASITOIDSP. 1. Trophic Level 3 Trophic. Level 2 Herbivore SP. 2. Herbivore SP. 1. Herbivore SP. 1. Trophic Level 1. Host plant associated plants. Associated herbivores. Figure 2.7 Semiochemically mediated interactions among members of 4 trophic levels, based on a composite of examples in the literature. Arrows are placed against the responding organism. Thick solid lines and solid arrows indicate attraction of a stimulus, e.g. 1, 4, 11, 24. Thin solid lines and open arrows indicate repulsion, e.g. 3, 13, 17, 26. Thin dashed lines show indirect effects such as interference with another response, e.g. 2, 12, 19. From Price 1981. Also cause digestive and other metabolic problems in mammals. Many other kinds of compounds are discussed as active ingredients in plant-herbivore interactions in Rosenthal and Berenbaum, 1991, alkaloids, terpenoids, cyanogenic glycosides, cardenolides, coumarins and others. In Figure 2.7, Interaction 2, associated plants may play a role in attraction or repellency to the host plant. When host crucifers were planted in mixtures with non-food plants with aromatic bow, the crucifers were less attacked by flea beetles, Philotreta cruciferae, Tabonanen and Root 1972. This interference the authors called associational resistance. Associated plants may also act as attractants to adults by providing nectar, pollen or chemical cues to herbivores, interaction 4, or they may be directly repellent to herbivores, interaction 5. Interactions within trophic level 2 include attracting conspecifics with sex pheromones, interaction 6, or repellency through epidictic pheromones, interaction 7. These kinds of interactions may be interspecific, as when bark beetle attacks attract other species, interaction 8, or in other cases repel attack by other species, interaction 9. Associated herbivores may have similar effects, as in interactions 8 and 9, interaction 10. Interactions among trophic levels 2 and 3 include influences from all 3 trophic levels. Many cases have been found in which a predator or parasitoid is attracted to herbivore by a chiromone, interaction 11. As was stated earlier in this chapter, Cardiochilus nigriceps finds its hosts, Heliothis equals Helicoverpa fluorescens, in response to 3 chemicals in the mandibular gland secretion of the host larva. Microplytis croceps Hymenoptera. Braconidae finds larvae of the moths Heliothis Z and Heliothis fluorescens by a chemical in larval Fras 13-methylhentriocontane. Scales left by ovipositing moths provide chiromones for the egg parasitoid Trichogramma evanescens, Hymenoptera, Trichogrammatidae. Similar interactions involve predators, such as larvae of Chrysoperlicarnia, Neuroptera, Chrysopode being attracted to scales from adults of Heliothis Z which stimulate egg predation, and bark beetle pheromones are strongly attractive to specialist predatory clarid beetles. Chemicals from a non-target herbivore may also interfere with host searching by natural enemies, interaction 12. Of course, herbivores have their own defenses, many of them chemical, e.g. Evans and Schmidt 1990. Interaction 13 is illustrated by the everted osmeterium of papilio butterflies, which releases foul-smelling odors, like rancid butter, which are butyric acid derivatives, detected even by humans. Sawfly larvae feeding on conifers store resin from the host plant in diverticula of the gut and regurgitate it when attacked. Many plant toxins are sequestered by herbivores, such as cardiac glycosides by milkweed-feeding insects. Aphids release alarm pheromones which attract ants, and the ants repel parasitoids and predators. The ants may well repel enemies of other species on the same plant, providing an example similar to interaction 12. Herbivores may also reduce chiromonal cues by cutting and dropping leaves on which they have fed, or by feeding nocturnally and leaving the feeding site during the day when tachnid parasitoids are active. Interaction 14. Enemies may also be attracted to herbivores associated with the target species, or their products, such as honeydew. Interaction 15. Interactions among trophic levels 1 and 3 involve plant hosts which are attractive or repellent to natural enemies, or associated plants which act in a similar way. Chemical signals may involve chiromones from plants directly, chiromones from the herbivore, such as frass which contains plant constituents, cinnamones such as floral fragrances, or apnumones from such things as fermentation products from rotting plant parts. An example of interaction 16 concerns corn plants which contain trichosane, which also appears on the eggs of the corn earworm, Heliothes C, and this chemical becomes a chiromone for the egg parasitoid Trichogramma evanescens, as also mentioned under interaction 11. A similar case concerns heptanoic acid present in potatoes which appears in the frass of the potato tuberworm, Thorimea operculella, which then acts as a chiromone for the parasitoid, Orgulus lepidus. Plants may also be repellent to natural enemies of herbivores. Interaction 17, as when the odor of pines is repellent to the tachnid parasitoid, Eucarcelia rutila, during its preovipositional period, while oaks are attractive, housing aphids producing honeydew. When the parasitoid is ready to lay eggs, pine becomes attractive, on which it finds its major host, the pine looper, Bupulus pinearius lepidoptera, Geometridae. Trichome exudates may also bar small parasitoids which become entangled, as with small wasps attacking the tobacco hornworm, Manduca sexta, on tobacco, and they are commonly effective on other solanaceous plants. Interaction 18 concerns associated plants acting as attractions to natural enemies on target plants. Commonly, flowering plants in orchards are attractive to parasitoids, such that codling moth, Sedia pominella, and eastern tent caterpillar, Mollicosoma americanum, were attacked more by the ichneummonid, Edoplectus conquisitor, when nectar was available. Conversely, associated plants may interfere with searching by natural enemies and reduce attack rates, as in attacks on the larch sawfly, Christophora ericksoni, when it occurred in a mixture of other trees and forest shrubs and herbs, with reductions of parasitism from around 86% to 10-13%. Interaction 19. Interactions within the third trophic level follow the same sort as in trophic level 2, interactions 20 to 23. For example, trail odors of litter-dwelling parasitoids become repellent to the same and other species, and associated parasitoids may well have similar effects. At the fourth trophic level, hyperparasitoids may be attracted to the presence of the herbivore. Interaction 24. For example, the ichneummonid hyperparasitoid, Euceros frigidus, lays eggs about 10 centimeters from a colony of sawfly larvae, and proximal to the tree stem, so that a sawfly colony passes as they feed on needles sequentially down the shoot. Euceros eggs hatch, and the planodile larvae crawl onto sawflies, becoming phoretic, and wait, maybe weeks, until a primary parasitoid attacks, after which the Euceros planidium becomes parasitic on the primary parasitoid. See Chapter 8 for more information. In interaction 25, a hyperparasitoid may be attracted by odors of the primary parasitoid. For example, the odors of female Diarrhetiella rapi which attacks aphids on crucifers are attractive to the hyperparasitoid, Cherops brassicae, which can then find parasitized aphids with wasp larvae it can attack. See figures 1.4 and 12.1 for a food web based on crucifer plant species. Finally, natural enemies at the third trophic level are commonly defended chemically against attack from the fourth trophic level. Ladybird beetles of many species contain toxins in the hemolymph which are repellent to would-be predators. Bloom 1981, and some pupae have a snipping device between abdominal segments, which closes and bites when a larva is attacked. Figure 2.8, Schroeder et al. 1998, Eisner 2003. Predatory bombardier beetles, Aptlina medbrachinus explodens and B. crepitans, explosively eject a hot mixture based on hydroquinone sand and oxidizing agent, hydrogen peroxide, which is highly effective against vertebrate and invertebrate predators alike. Eisner 1970, 2003. Thefrican bombardier beetle, Stenoptinus insignis, is brightly colored, aposematic conspicuously marked, and flightless, all providing a cue to a very toxic spray. Figure 2.9. The impressive diversity of chemical information transmitted and received illustrates how each species can communicate in a specialized way, perhaps unique in some cases. Although a local airspace may be redolent with both from community members, each signal has a source with a more or less specialized receiver. Communication can be precise and effective. However, such signals can be decoded and utilized with surreptitious intent, as with clarid predators of bark beetles using pheromones to find their prey. We observe, as increasingly these kinds of revelations are made, that natural selection has resulted from almost endless kinds of chemically mediated interactions, and that there is probably no end to the ingenuity of nature still to be discovered. Of course, an equally diverse scenario of interactions through trophic levels could be developed for physical factors concerned with defense and attack. Some of these are discussed in Chapter 4 on plant defense and herbivore feeding, and in Chapter 7 on prey and predator interrelationships. 2.8 Reproductive behavior. A place to live also involves a place to breed, and the whole reproductive process from finding mates to oviposition in appropriate places, which is treated here as the final aspect of behavior to be discussed in this chapter. Insect reproductive behavior was reviewed comprehensively from an evolutionary perspective for the first time by Thornhill and Alcock, 1983, The Evolution of Insect Mating Systems. The book remains a major source of information, fascination, and inspiration. While the field has advanced significantly since that date, the book provides a systematic approach to this large subject, from which we have selected some major aspects to discuss in this chapter. We need to start about 150 years ago with Darwin's 1859 creation of the concept of sexual selection before we move on to the various methods of mate selection, mating strategies and finally parental investment. 2.8.1 Sexual selection. In The Origin of Species, Darwin, 1859, page 88, introduced the concept of sexual selection as a subset of natural selection. This depends, not on the struggle for existence, but on the struggle between the males for possession of females. The result is not death to the unsuccessful competitor, but few or no offspring. Sexual selection is, therefore, less rigorous than natural selection. Generally, the most vigorous males, those which are best fitted for their places in nature, will leave most progeny. But, in many cases, Figure 2.8 Defense of the ladybird beetle pupa, Cyclonata sanguinea, formed by pincer-like gaps between abdominal segments, see arrows in frame A. When disturbed from behind by a predator, or in this case a bristle, the pupa flips up, closing the gap and biting the aggressor. Photographs by Thomas Eisner, Cornell University. From Schroeder et al., 1998, and Eisner 2003. Copyright 1998 National Academy of Sciences, USA. See color plate section. Victory will depend not on general vigor, but on having special weapons, confined to the male sex. The hornless stag or a spurless cock would have a poor chance of leaving offspring. Darwin may just as easily have mentioned the stag beetle with the male's large mandibles with which they joust, or the spurs and horns of dung beetles and other scarabs used in displacing opposing males. Indeed, when Darwin, 1871, greatly expanded on his theory in the descent of man and selection in relation to sex, he noted many examples in which male beetles exhibited greatly enlarged mandibles or horns, figures 2.10, 2.11. Of course, when we consider fitness of individuals, then natural selection and sexual selection become as one, because inability to breed may not kill, but leaving no progeny is genetic death, failure to impart progeny or genes to the next generation. However, the term sexual selection is very useful in emphasizing differences in male and female form and reproductive behavior. The concept was expanded in Darwin, 1871, to include two forms, each emphasizing the role of one sex. 1. The competition among males for mating with females, called intersexual selection by Huxley, 1938, in which females are likely to be passive. 2. The active choice by females of males which appear to be high-quality mates, based on size, color, offensive weapons or vigor, named epigamic selection by Huxley. That males differ greatly in reproductive success, while females are more predictably successful, was elegantly revealed in now famous experiments by Figure 2.9 The African bombardier beetle blasts off to repel an attack by a pair of forceps which simulated the mandibular grasp of a would-be predator. The blast is directional so that it is projected forward in A, sideways in B, and downwards when the hind leg is pinched. See photographs by Thomas Eisner and Daniel Inez Hansley. Caption for Figure 2.9 Kant. Cornell University. From Eisner and Inez Hansley 1999, and Eisner 2003. A reprinted by permission of the publisher from For Love of Insects by Thomas Eisner, p. 31. Cambridge, Massachusetts. The Belknap Press of Harvard University Press. Copyright #2003 by the President and Fellows of Harvard College. B&C Copyright 1999 National Academy of Sciences, USA. See color plate section. Figure 2.10 Illustrations of dung and rhinoceros beetles, in Darwin's 1871 book on the descent of man and selection in relation to sex. Males are on the left, showing large horns, extensions from the head and thorax, and females are on the right with no such fighting equipment. Bateman, 1948. Using adult fruit flies, Drosophila melanogaster, in which each had a distinctive dominant marker gene, which would show up in the progeny they produced, Bateman mixed males and females for 3 or 4 days, and measured their success in competition with males for mates and access to females. Males showed much greater variation in reproductive success than females. Many males did not mate even once, 21%, while other males mated with as many as 4 females, figure 2.12, and some males produced more progeny than others. By contrast, the large majority of females mated once or twice, 96%, few more than this, and there was less. Figure 2.11 The stag beetle, Chiasognathus grantee, from southern Chile illustrated in Darwin 1871, showing the male with enormous mandibles and the female with normal-sized mandibles. The same species is illustrated in a defensive position on a tree trunk on which a fight will result in one of the males being displaced and falling to the ground. From Linsenmayer 1972. Berofflies. WITH. 0, 1, 2, 3, OR4. ATS. 012. 246 Number of mates. 34. Males. 108.0604020 females. BEROFO. FFSP Re NG 100. 50. 1 2 3 1 2 females males 3. Figure 2.12 Results from Bateman's 1948 experiments on the variation in male and female mating success in Drosophila melanogaster. Many males have no mates, whereas almost all females mate at least once. Some males had 3 or 4 mates whereas most females had only 1 or 2 mates. Below, the difference in the number of offspring produced by 3 females caged with 3 males is much less variable than among the 3 males. The spread of reproductive success among females is about 40 progeny, but the range among males reaches 80 progeny. Modified from Thornhill and Alcock 1983. Variation in the number of offspring produced by females than by males. Bateman did not examine the reasons for this differential breeding success among males, but it may well have related to male size, vigor, or the love songs with which they courted females. Hoykala 2006, Drosophilus and Claridge 2006. More conspicuously differentiated males and females than in Drosophila melanogaster reveal clearly the winners and losers resulting from jousts for access to females. Horned males and hornless females are observed commonly in beetles, as well as enlarged mandibles of males, stag beetles, Lucanidae, rhinoceros beetles, Scarabaeidae, Dynastinae, and dung beetles, Scarabaeidae, Scarabaeinae are well-known examples. Males assemble at places where females visit and fight for access to females. Such places may be sap flows, shoots or branches, or burrows in plants, or in dung beetles' subterranean tunnels in which females are found. Emlyn 2000a, Emlyn and Nijhout 2000. Although the resources in question vary considerably from species to species, they share one key characteristic: they all tend to occur in discrete, readily defendable patches. Males with enlarged weapons are able to gain disproportionate access to these contested sites, and consequently, gain disproportionate access to females. Emlyn 2000a, p. 404. In this context, selection will result in larger males with larger horns, until the benefits are outstripped by costs. Such trade-offs have become of great interest in the adaptive radiation of dung beetles in the genus Onthophagus, e.g. Emlyn 2000a, Mochek and Nijhout 2004, Parzer and Mochek 2008. Although large males win fights, small males may adopt alternative mating strategies, as we shall see later in this chapter. Sexual selection will run as a theme throughout the remainder of this chapter. A summary of the results of sexual selection reveals the diversity of behaviors involved. Table 2.4. 2.8.2 Finding mates. Two central questions relating to sexual selection posed by Darwin, 1859, are: first, why males and females of the same species differ from one another, with males showing more exaggerated morphological phenotypes than those observed in females, and second, why males of closely related species show much greater differences in morphology and behavior than females in these species. Schuster and Wade 2003. Clearly, males in most species have to find females, and then compete with other males for access, both activities tending to select for species' morphological traits, including enlarged antennae for detecting a female's pheromone, and enlarged appendages or horns for fighting. However, in many cases males have evolved to find mates as early as possible, thereby avoiding much competition. Thornhill and Alcock 1983. Males are known to search emergent sites when they can mate with emerging virgin females. Examples include bees, wasps, ants, butterflies and beetles. Another strategy is seen in males that find females where the females oviposit, as in scatophagid flies which mate on dung, and many fruit flies in the family Tephrididae, which mate on the fruits into which females oviposit. In other species males have gone to the extreme of detecting virgin females before they emerge as fully mature adults, with forced copulation resulting. Females may be more active in finding mates in many species where mating occurs at a site away from emergent sites, oviposition sites and food. Females may fly into groups or swarms of males located at landmarks, such as shrubs or trees, or hilltops. Such species include bees, ants, butterflies, midges, mosquitoes and other flies. Hilltopping is a common mating strategy for males and females in which mostly males fly to prominent topographical features and mate with virgin females as they fly through the area. This mate rendezvous hypothesis, Alcock and Dodson 2008, predicts that 1. Most individuals at hilltops are males which gain access to several females and mate with them, but females will be less abundant because they mate once. 2. Females that go to hilltops are likely to be virgins while mated females will be more active at feeding and oviposition sites. Just on one mountain in Arizona, Alcock and Dodson 2008 recorded 6 families of Diptera, 6 families of Lepidoptera, 5 families of Hymenoptera and 1 beetle family, Cerambicidae. Some species males were territorial, driving away others, but different species had males that simply patrolled hilltops without aggressive behavior. Hilltopping appears to be a rewarding strategy where populations are sparse, so that mates would be hard to find. For example, of the 6 fly families listed from hilltops by Alcock and Dodson 2008, 4 were parasitic groups: the bot and warble flies, Cuterbridii and Estridae, tachnid parasitoids and bombylids, which are parasitic on other insects such as bees or predaceous on grasshopper eggs. None of the host species would provide dense and predictable resources worth defending territorially, and females of parasitic species disperse widely in search of hosts, so a rewarding strategy appears to be to mate first at a hilltop site, followed by the lonesome search for hosts. Table 2.4 The products of sexual selection in animals in general. Not all products have been observed in insects. Intrasexual selection. Precopulatory competition for access to potential mates. 1. Skill in mate location. 2. Production of effective mate attracting signals. 3. Aggressive competence in the defense of mates and territories. 4. Capacity to avoid damaging interactions with rivals. Postcopulatory competition for access to eggs. 1. Mate concealment. 2. Mate guarding. 3. Ability to find and take protected mate from original partner. 4. Ability of sperm to displace competitor. Attributes that make opposite sex attractive to discriminating sex. 1. Attractive courtship behavior. 2. Morphological characters considered attractive by opposite sex. 3. Material benefit attractive to opposite sex from Thornhill and Alcock 1983. Reprinted by permission of the publisher from The Evolution of Insect Mating Systems by Randy Thornhill and John Alcock, p. 74. Cambridge, Massachusetts. Harvard University Press. Copyright #1983 by the President and Fellows of Harvard College. Assemblages of males, independent of food or oviposition sites, are called leks. Males aggregate at one spot, set up small adjacent territories and attract females, and the females may select a male for mating and then depart. Males may then mate again with additional females, benefiting from the aggregate attraction of the assembled males. Figure 2.13. This mating system illustrates select polygyny, in which males usually mate more than once, whereas females mate only once. Male tarantula hawk wasps, Hemipepsis ustulata, also engage in polygyny in the desert of the southwest United States, where they set up territories along ridgetops inhabited by palo verde trees, creosote bushes or jojoba shrubs, which provide conspicuous perches. Territoriality takes many forms among insect species. A territory is defined as any defended area where resources are moderately concentrated, relatively patchy, but stable in time and space. A male may increase access to females by becoming territorial. Males may set up a territory where females pass through in order to oviposit, or where females are attracted to oviposition sites or food. See Price 1997 for territory types. The first case of females being intercepted as they fly to oviposition sites is illustrated by the peacock butterfly, Anachus eo, in which males place their territories in a field through which females flew in order to oviposit on stinging nettles beyond. Figure 2.14. In the corner of the field surrounded by a hedge on two sides females converged, and the male in territory 1 gained access to 4 females. Another male with territory 2 by the hedge has 3 females traverse his territory, but territories 3 and 4, away from the hedge, were less successful. This strategy avoids competition at oviposition and feeding sites, but evidently results in large differences in success among males. A more predictable ploy, but one with a greater competitive risk, is to defend an oviposition site, as does the speckled wood butterfly, Pararge agria, Davies 1978. Males defended sunspots on the forest floor, which were limiting, so only 60% of males occupied territories. Females were attracted to sunspots because the grass host plants to larvae grow in the brighter areas of the forest floor. Males without territories patrolled the tree canopies searching for mates, but were much less successful than the territory holders. The complexity of a landscape may be diminished by chemical lures, pheromones, which are sex attractants, usually released by females, which attract males, followed by copulation, Roloffs 1981, Carday, and Baker 1984. Males fly up plumes of odors, gradually orienting to the releasing female, often aided by enlarged antennae relative to those of the female, as in Saturniid moths, Diprionid sawflies and Figure 2.13. A lack of Hawaiian fruit flies, Drosophila heteronera, on a tree fern. The fern provides only an assembly area, with no oviposition sites, food or emerging females. Males joust for a small territory in the lek, at front right. A female visits the lek seeking a mate, top right, and a copulating pair is illustrated on the left. Drawing by L. S. Kimsey. From Thornhill and Alcock 1983. Scarabaeid beetles. In the giant silkworm moths, Saturniidae, marked males of the Cecropia moth, Hyalophora cecropia, are known to fly many miles overnight to a female, up to 11 miles in one case, although how far the female's pheromone plume was involved was not investigated. Waldbauer 1996. Promethea moths flew 9 miles in 3 days, and even 23 miles in 3 days, to find a virgin female in a trap. With such effective discovery of mates over long distances, it is possible for species to persist at very low densities and in highly patchy environments. 2.8.3 Choosing mates. Once a mate has been located, there may be almost no courtship behavior before copulation, as in many sawflies, or courtship may be extravagant. Generally, males appear to be the amorous sex, because they initiate courtship more often than females. Females, on the other hand, appear to be more selective than males because they tend to mate only once. The quality of the father of her progeny is critical. However, because each mating involves one male and one female, the average number of mates for males and females must be equal. That is, there can be no sex difference in average promiscuity between the sexes. Wade and Schuster 2002. Sex differences in mating behavior are often attributed to differences in energetic investment in gametes. Sperm are cheap, eggs are expensive. With eggs relatively large and costly in allocation of resources, it becomes important for females to choose males which are likely to father strong and attractive male progeny, and large and fecund female progeny will amplify a female's fitness relative to an unselective female. However, a simpler explanation for these differences is, as Bateman 1948 showed in Drosophila, that the correlation between mate numbers and offspring numbers is usually much greater for males than it is for females. Male fitness increases linearly with Figure 2.14. The location of male territories of the peacock butterfly, in a field through which females flew to reach oviposition sites. Note differences in success among males in acquiring females based on territory positions. From Baker 1972a. Reprinted with permission from Blackwell Publishing. Increasing mate numbers, whereas female fitness increases less rapidly, and may even decrease with multiple mating. Strong selection on males to increase mate numbers and on females to minimize them provides an explanation for why males of many species may attempt to court or copulate with mated females or even with males, while females may kick away a male, fail to adopt a copulatory position or simply fly away. Thornhill and Alcock 1983. This is epigamic selection at work. Courtship takes many forms. Acoustic signaling is a major method for attracting mates, with stridulation of crickets, katydids, grasshoppers and cicadas commonly experienced by the naturalist, as well as females in these groups. But the fact is that much acoustical and vibrational communication passes unnoticed by the human ear. Like chemical communication, sounds and vibrations made by small insects escaped our attention for many decades. In a wonderful coverage of the literature, Drosophilus and Claridge 2006 included many groups of smaller insects, lacewings and other neuropterids, Drosophila flies, stoneflies, planthoppers, leafhoppers, heteropteran bugs, treehoppers, psyllids, whiteflies and bees. Not only is mate location involved with vibrations, but membracids recruit to new feeding sites, gerrids and others locate prey, purgid sawflies use vibrations for group cohesion, ichneumonids, braconids and other parasitoids use them for host location, and some ants recruit to food sources, Virant Doberlet et al. 2006. Also, spiders and scorpions are well known to use vibrations in prey and mate location. Courtship also includes nuptial gifts of food from males to females in many species. The male can display his competence as a hunter and/or provider of food, and the female can make a choice as to whether the gift is adequate. Male gifts may contribute to female fitness by providing a significant meal which enhances her fecundity, and it reduces her risks while hunting for food, and in so doing it promotes male fitness as well. Thornhill and Gwynn 1986. For example, male hanging flies, such as Hylobodicus apicalus, a member of the scorpionfly order Macoptera, catch insect prey and attract a female. If she accepts the gift she starts feeding and the male is able to copulate, figure 2.15. In katydids the male delivers a large spermatophore to the female consisting of two parts, one is an ampulla containing sperm, and the other is a food item, free of sperm, called the spermatophylax, figure 2.16. After mating the female eats the spermatophylax while insemination occurs, and then she eats the empty ampulla, acquiring a considerable contribution to egg production from the male, and also acquisition of substances which induce a 4-day-long non-receptive period, ensuring the male's paternity. Thornhill and Gwynn 1986. The parental investment of the male in the form of the spermatophore results in an increase in egg size and fecundity of mated females. Some male katydids provide such rich gifts, spermatophores of 25 to 40% of their body weight, that females actually compete aggressively for males. Figure 2.15. A pair of hanging flies, Hylobodicus apicalus Macoptera, Bitacidae, with the male on the left and the female on the right. The male has captured a blowfly, attracted the female, and has presented her with this nuptial gift which she is consuming. The male can then copulate with the female. As shown, the female's fecundity is increased by the gift and her probability of survival is improved by the reduced need for foraging. Both male and female reproductive success is improved. From Thornhill 1980, see color plate section. And males select large fecund females. In the Mormon cricket, Anabras simplex, actually a katydid in the family Tetagoniidae, this reversal of the sex roles is observed. Gwynn 1981, 1984. Female choice of mates is an important step in bringing mating to fruition, and females make important decisions because, as stated earlier, they may mate only once and their investment in progeny is large relative to that of males. So females make many decisions using a variety of cues to evaluate males. Female cockroaches of Nophoda prefer the pheromone of dominant males. Other females may accept the most unusual males of a group, the rare male advantage in Drosophila for example, in which females are more likely to mate with males with the less common genotypes. Thornhill and Alcock 1983 discuss many methods by which females reject males, prior to, during or after copulation. One intriguing idea, formulated by Eberhard 1985, is the female choice hypothesis. This argues that females choose males of their own species on the basis of the male's genitalia. Males with favored genitalic morphologies sire more offspring than others. Eberhard 1985, p. 7.0. Then sexual selection on male genitalia by females results in rapid diversifying evolution. Eberhard 1997 expands on this thesis, emphasizing cryptic female choice and listing the many mechanisms by which this is achieved. Indeed, it has been long remarked that male genitalia in insects show incredible variety among even closely related species, and often genitalic characters are diagnostic of species differences. A recent example of extreme variation in male genitalia is from Bembidian carabid beetles on the Hawaiian Islands, with 23 species showing dramatic variation, including exaggerated male mating organs, heterotrophy, on the male adeagle flagellar complex, Liebherr 2008, his figures 3 and 4. The flagellum is involved with placing the male's spermatophore into the female's long spermathecal duct during copulation. The intimate evolutionary association of an intromittent male structure with the corresponding female. Figure 2.16. Just after mating, a female katydid, Requena verticalis, has a large spermatophore attached to the base of her ovipositor by the male top. The female bends down to grasp the nutritious spermatophylax, middle, and consumes it, bottom. The ampulla with the sperm remains in place, releasing sperm, and when this is complete the ampulla is also eaten. The female gains a major donation of food from the male, resulting in increased egg size and fecundity. Adapted from Thornhill and Gwynn 1986. See color plate section. Receptive structure strongly supports the action of sexual selection during diversification of these beetles. Liebherr 2008, p. 7.2. This selection may result during female choice of superior males, or sexual conflict in which females are resistant to mating, which selects for heterotrophy. One can easily imagine that in local populations selection may shift populations in different directions involving genitalic compatibility, resulting in reproductive isolation between populations, speciation and adaptive radiation, in such heterogeneous landscapes as provided by the Hawaiian Islands. 2.8.4 Mating systems and strategies. A mating system may be defined as a specific pattern of male-female associations. The term, mating system, of a population refers to the general strategy employed in obtaining mates. It encompasses such features as I. The number of mates acquired, E. The manner of mate acquisition, E. The presence and characteristics of any pair bonds, and IV. The patterns of parental care provided by each sex. Emlyn and Oring 1977, p. 222. Thus, much of the preceding parts of this chapter come under the umbrella of mating systems. Schuster and Wade 2003, page 36, summarized Emlyn and Oring's 1977 ecological classification of animal mating systems as follows. 1. Males compete with one another for access to females. 2. Like competition for scarce resources, male reproduction is limited by the spatial and temporal availability of sexually receptive females. 3. The intensity of sexual selection depends on the rarity of receptive females in relation to the abundance of competing males. 4. Sexual selection favors male attributes which permit their bearers to find and monopolize their mates. 5. Ecological constraints on male monopolization attempts lead to a species-specific pattern of male-female associations, called a mating system. The intensity of competition for mates, or the intensity of sexual selection, is often estimated using the operational sex ratio, which is the ratio of adult males to adult females. However, because this ratio can overestimate the success of certain individuals, a better estimator of the intensity of sexual selection is the average number of mates per mating male, a value proportional to both the variance in male mating success and the intensity of sexual selection. Schuster and Wade 2003, Wade and Schuster 2004. This view can result in a classification of mating systems based on male and female mate numbers. Schuster and Wade 2003, Table 2.5. And we have Schuster and Wade to thank for clarifying and simplifying the glossary of terms for mating systems. Differences in mating systems depend on several factors, making males or females more or less accessible. Where both sexes care for young, monogamy is likely to prevail, as in Microphorus beetles, Sylphidae, in which both sexes make a nest under a buried carcass and feed their young in the nest. Monogamy appears to be an ancestral trait for bees, wasps and ants, the eusocial insects. Hughes et al. 2008. Termites may also form persistent pairs. The term polygyny. Schuster and Wade apply exclusively where females mate with only one male in their lives, but males may mate with multiple females. This mating system may represent many territorial species, as with territorial hilltoppers such as tarantula hawk wasps mentioned earlier, and the territorial butterflies and lecking species such as Drosophila heteronera, figure 2.13. Polygynandry esoust only is stated in table 2.5, and Schuster and Waddle list only scorpionflies and water striders as insect examples. In the scorpionflies, for example, a male may feed and copulate with several females. Onda is unable to guard females while hunting for prey, opening opportunities for a female to gain more nuptial gifts and sperm from other males. In polygamy, males and females mate more than once, and males cannot protect females from other males over the longer term. Therefore, conflict among males for females, and intersexual conflict with females rejecting males, may occur as in Drosophila and in damselflies. Sperm competition, discussed later in this chapter, is well known in these insect groups. Polyandrogyny is the converse of polygynandry, and refers to mating systems in which females demonstrate the more variable number of mates. We would expect this mating system to prevail under the unusual circumstance in insects of male parental care in which males are constrained in mating because protecting young is paramount, while females are free to mate with several males. An example is the giant water bugs Elastomatidae, Abetus, Belastoma and Lethoceras discussed in Chapter 10 on life histories. Polyandry involves males that mate for life with a single female, but females live much longer, or males become devoted to full-time parental care after one mating. These cases are rare in all animals, but one example concerns praying mantids, order Mantodia, and some spiders in which the female eats the male during or immediately after copulation. Understandably, this classification of mating systems simplifies the diversity seen in nature, and Schuster and Wade provide a more comprehensive survey of major categories and subcategories. One additional strategy is practiced by parthenogenetic individuals or species, some of which do not mate at all. In any one species of insect, one major strategy may appear to be well represented, but alternative mating strategies often exist. For example, in many beetles the big males win competitive bouts with smaller males. But small males are unlikely to live until another breeding season, so selection will probably promote an alternative strategy. These alternatives may employ surreptitious behavior and mating, leading to the epithet of sneaky fellow, or something more risque, for these males. For example, in the dung beetle, Onthophagus acuminatus, there is a bimodal distribution of horn lengths, with small males hornless and large males well equipped with horns, figure 2.17, Emlyn 2000a, B, Emlyn N. Nijhout 2000. The large males guard burrows under dung, while females burrow below and provision larval cells with dung. Small, sneaking males adopt an alternative strategy involving a side burrow, circumventing the large male, and gaining access to the female. Onthophagus taurus exhibits even stronger horn dimorphism than O. acuminatus, Stern and Emlyn 1999. With both morphs of Onthophagus able to mate successfully, presumably selection will maintain this dimorphism indefinitely, if the success of the strategies is more or less matched. Another case of an alternative mating strategy was described earlier for the speckled wood butterfly, in which some males established territories, while others cruise the tree canopy in search of females. Thornhill 1981 described alternative mating strategies for the scorpionflies, Panorpa species Macoptera, Panorpidae, and these are listed in order of likely success in acquiring copulations. Some males defended dead insects which were alluring to receptive females. Others secreted saliva onto leaves, providing a nuptial gift for the occasional female. A third strategy was to force copulation without a gift of any sort. The males made the best strategic choice available to them, and were opportunistic about the strategy they adopted, a conditional strategy depending on available resources defined by the competitive milieu in which they found themselves. Grasshopper males also show alternative approaches, and these alternatives may change in individuals over time, Shelley and Greenfield 1985, or in other species some may produce large spermatophores while others attempt to force copulations, Bolovsky et al. 1996. Large males of water striders, Geras remigus, are able to swim in fast-flowing water with more food and mate with large females, but smaller males swim in slower water, Rubenstein 1984. Some species of fig wasp have twomel morphs. Grief 1995. One type mates with fig and never leaves, while the other morph can disperse before mating and finds mates on leaf-sore fruits. The variety of alternative mating strategies among species is extraordinary. B. 2.25. A. 0. 0.25. 0.75. 1.25. 0.5. 1.5 2.5 2.75. Body size H or N. Leing th. M. Horn length. Body size. N B B 2.6